Greetings, students. So today we are going to begin looking at population genetics. And what population genetics does is it sort of bridges the ideas of evolution and genetics. So Charles Darwin, he sort of was focused on what we now call macroevolution, which are these large changes that take place over long periods of time. And we saw lots of examples of that, how whales evolved from, four-legged animals that lived on land, transitions between different species, that kind of thing. What we're going to be focusing on now in this chapter, though, is microevolution. Microevolution refers to changes in allele frequencies in a population over many generations. And in fact, this is the more modern definition of evolution. If somebody says, what does evolution mean? The technical definition is it is changes in allele frequencies in a population over time. So an example would be if you had this beetle population with some green and some brown beetles, and let's suppose in one generation you had 75% green beetles, 25% brown, but then in the next generation there was only 71% green, 29% brown. Well, you would say that the allele frequencies have changed and therefore evolution has occurred. So we're going to get into more details of that soon. So before we do, though, we need to talk a little bit about genetic variation. Now, we've talked a fair amount about genetic variation already and the sources of that. And so you definitely want to take some time to sort of review that. So basically the main sources of genetic variation, one, mutation. That's sort of the ultimate source of all genetic variation. And we've seen lots of examples of how new alleles can be formed through changes in or errors in DNA replication, whether it's a point mutation. We've seen how you can have whole chromosomes be changed with a deletion or an insertion or a translocation during errors in mitosis and meiosis. So we've seen lots of examples over the year of where this genetic variation comes from. Another source of genetic variation, rapid reproduction. Things that reproduce more quickly will actually evolve more quickly, and the reason why is every time that genetic material is copied and passed to the next generation, you have more opportunity for these random mutations to occur. So bacteria who reproduce several times a day have more opportunity to mutate and change than, say, humans who reproduce every, when they're at least 29 years of age and in a committed relationship and financially secure. So that makes a big difference, is how long it takes to actually propagate that genetic material. Finally, sexual reproduction is a big source of genetic variation as well. We saw that crossing over during prophase 1 of meiosis, where those homologous chromosomes exchange little bits of DNA, that creates new combinations of genes that didn't necessarily exist before. Independent assortment. When those chromosomes line up down the center of the cell in metaphase 1 of meiosis, those homologous pairs, if big A is with big B, or if big A is with little b, that again creates new combinations of genes. And then just random fertilization, since meiosis produces this widely varied group of sperm and egg cells, the particular sperm that joins with a particular egg is going to create a particular set of genes that, again, are going to be different from other combinations. And that's why you are very different from your brother or sister, even though both of you came from the same genetic material. So all of that should be reviewed, and be sure you take some time to kind of review that in your book if any of what I just said is new to you. Okay, so new in this chapter is how do we maintain this genetic variation? So what are some ways in nature that this genetic variation is maintained? So one that we've sort of talked about, but not really in this context, is just being diploid. So if you are diploid for a trait, essentially that's hiding recessive alleles from natural selection. The phenotype is not going to be expressed in the heterozygotes, and that's why and that allows this sort of recessive allele to sort of just kind of linger in the genetic material and not be exposed to the rigors of natural selection. And so that's why oftentimes, not always, but oftentimes, negative mutations, like harmful things like diseases and conditions, oftentimes they're recessive because that sort of protects them from the cold hand of natural selection and prevents them from being eliminated. They only create a problem when those two recessives come together in the homozygous recessive condition and then are exposed to natural selection. Another way to maintain genetic variation is what is called heterozygous advantage. This is where a heterozygote has a higher fitness than either homozygote. There is a very interesting and fascinating story, example of this, which is sickle cell anemia. If we're doing this online, then I would really urge you to click and watch. This is a fairly long video, but it's really pretty interesting and worth it, I think. Typically I do show this in class. But the gist is that individuals who have sickle cell anemia, typically their ancestors came from parts of the world where malaria is present. And so people who descended from people who lived in certain parts of Africa are more at risk of sickle cell anemia. And the reason why is if you have two recessive traits, versions of this trait, then you are going to end up with sickle cell anemia, which is a harmful variation that makes life difficult. If, however, you have homozygous dominant, you have two copies of the dominant trait, then you're not going to have sickle cell anemia, but it turns out that you are more at risk of being infected with malaria. And so it turns out that if you're heterozygote, you have an advantage over either homozygote, because if you're a heterozygote, you don't have sickle cell anemia, but you're also more resistant to malaria. And so in areas where malaria is a problem, that dominant allele, that recessive allele sticks around more than you'd expect, because in the heterozygotes it gives an advantage that it wouldn't give in the absence of malaria. So again, this video is very cool, kind of shows that, and I would encourage you to watch it. All right, one final way to maintain genetic variation is frequency-dependent selection. So here, the fitness of the phenotype depends on how common it is in the population. So an example of this, there's these scale-eating fish. This just seems cruel. You can see in this picture, here's like a nice little chillin' fish. And then here you've got a fish, this is the scale-eating fish, and its mouth is kind of leaning to the left. But then you also have a variation where the mouth leans to the right. And so which of these has the advantage depends on how common they are in the population. So for example, if I'm just a fish, just chilling, and all of a sudden this thing comes up next to me and like nips me, and then another one comes up and nips me, and then another one, and another one, I'm going to realize like, man, there's a lot of these guys coming at me from this side. And so I'm going to start to be a little more alert to this side and defend myself a little bit. So if you are of the opposite type, then all of a sudden, while I'm guarding against all these guys over here, you can come out and do this sneak attack and out of nowhere and take me out and get your little scale to eat. And so if you're rarer, then you actually have an advantage in this population. And so what happens is the frequency of the left mouth individuals, it kind of oscillates over time, because if it's common, then the right mouth has an advantage. But then if they go down in population and the right mouth becomes more common, then now the left mouth has an advantage, and so they rise up. But now the right mouth has an advantage, so the left mouth goes down, and so on. So we call this frequency-dependent selection. It depends on how common it is, whether or not it's advantageous. So those are all different ways that we can maintain genetic variation. I think we'll pause there, and in the next screencast we'll take a look at population genetics more specifically.